Hello, everyone, and welcome to today's webinar titled Keynote Presentation, The Virus, the Host, and the Battle for Survival, How Viruses Interact with the Host Ubiquitin Machinery to Promote Susceptibility and Resistance to Infection. This webinar is part of the 2021 Microbiology Virtual Week. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, just click on the Ask a Question box located on the far left of your screen. And as a reminder, the presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located on the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Ricardo Rajsbaum, an associate professor of the Department of Microbiology and Immunology at the University of Texas Medical Branch. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Rajsbaum, welcome. You may now begin your presentation.
Good morning, everybody. First of all, thank you so much for LabRoots for the invitation to give this talk and to be able to share with you some of the work that we are doing, which we think is very timely during this terrible COVID pandemic, to understand how different viruses are able to replicate and adapt to the host cells. So to start, upon virus infection, the host cell can recognize the virus infection via pattern recognition receptors that are expressed in different compartments in the cell, and upon recognition, trigger multiple signaling cascades that result in the expression of different cytokines. These cytokines have different functions during viral infection. One of the cytokines that they are particularly interested in is type 1 interferons, because they are well known to have antiviral effects. So these interferons can act by triggering their own signaling cascades through the receptor in neighboring cells, and this results in the expression of interferon-stimulated genes, or ISGs, which are proteins that can directly have antiviral effects by interacting with the viruses. But obviously, if this was perfect, we would never get infected. So viruses have evolved to express proteins that can antagonize these responses, and basically can antagonize each of the steps that I'm showing in this slide. But we're particularly interested on the interactions that happen between these viral antagonists and the host antiviral effectors that can lead to some evolutionary pressure to the virus and allow the virus to adapt and use these antiviral effects for their own advantage. So we are really interested in looking how these potentially antiviral factors can be utilized by the viruses for their own advantage. So what I'm going to tell you here, it's examples of different viruses that we have been studying that include the Nipah virus, Ebola virus, influenza, Zika, and I'll touch a little bit on SARS-CoV-2. So to zoom in a little bit more on these pathways, specifically the RIGI pathway, which is a pattern recognition receptor that can recognize viral RNA when viruses infect cells and trigger this specific signaling pathway. And what I want to focus here is on these kinases that are called TDK1 and IKK epsilon that can phosphorylate transcription factors IRF3 and IRF7 that translocate to the nucleus for production of type 1 interferons. So basically, these pathways have to be highly regulated so we do not have excessive inflammation. This would cause problems. If we have too much inflammation, even interferons can be bad if they are expressing high levels. So these post-translational modifications like phosphorylation can turn on and off these pathways. So another post-translational modification that we're interested in studying is ubiquitination, which is mostly studied for modification of proteins that are targeted to degradation by the proteasome. But ubiquitination has many other functions, and like phosphorylation, can regulate, activate, or deactivate these pathways. And there are different types of polyubiquitin chains that have different functions. So ubiquitin is a small protein. It's a 76 amino acid protein that the way it functions is that modifies proteins via its C-terminal glycine residues that covalently modify usually a lysine in a target protein. So these, in addition to this, since ubiquitin itself has 7 lysines, each of these lysines can be modified by another ubiquitin to form these polyubiquitin chains. This is important because depending on the polyubiquitin chain that it can be formed, like for example, one of the ones that is most studied is lysine 48 ubiquitin chains that are targeted to the proteasome for degradation, have that function, but there are these other chains that are less studied and have non-degradatory functions. So, and the way, why is it important? How does it work? Why is it that the chain, depending on what lysine is modified, the chain would be different? And really, is the topology of the chain. Is how here's an example of how when you have a lysine 48 linkage, you have one type of topology or shape of the chain, and when you have a lysine 63, you can have a different type of shape. And these would change how other proteins interact with the ubiquitinated protein. So in general, proteins that are going to lead to signaling cascades are recruited or interact with ubiquitinated proteins and require or have ubiquitin binding domains that is like a lock and a key. They have to match so they can interact and efficiently with high affinity and form complexes that lead to signaling cascades. So the ubiquitination process requires the activity of 3 enzymes, an E1 activating enzyme in the presence of the ATP as an energy source and ubiquitin, an E2 conjugase, and an E3 ligase. The E3 ligase is really important because it's the one that it's required for recognition of the substrate or the protein that is going to be ubiquitinated. So the E3 recognizes the E2 conjugase together with the substrate and transfers ubiquitin to form these polyubiquitin chains that will lead to a specific function depending on the type of ubiquitin. Can be degradation by the proteasome or many other functions depending on the polyubiquitin chain. There are about 600 E3 ligases that are encoded by the human genome. We are particularly interested on one of these families that is called tripartite motif or TRIM proteins. It's composed of over 70 members in humans and contain this specific domain, a ring, a B-box domain, and a colloid domain, and different TRIMs can contain different C-terminal regions. And why are we so interested in this TRIM family? The reason is because indeed in 2004 it was discovered that TRIM5 alpha could have antiviral activity against HIV. Specifically, the African green monkey and macaque TRIM5 alpha could restrict HIV. So since then, a lot of work was done on studying retroviruses and how multiple TRIMs could potentially inhibit directly or restrict these viruses. But it took a long time until really TRIMs took off, and other studies were looking at other forms, other types of regulation in terms of antiviral activity. TRIMs can have many other functions like cell differentiation, transcription regulation. They are ubiquitin ligases, so you would expect them to be involved in many functions. So really the role of TRIMs, there were a few studies that appeared to show that TRIMs could also regulate signaling pathways, but really the major discovery, I think it was in 2007 when Michael Agag discovered that TRIM25 could ubiquitinate RIGI to trigger that signaling pathway to induce interferon. Since then, a lot of studies have focused on how TRIMs can also regulate inflammatory pathways or interferon pathways. Another thing that can connect this with antiviral responses is that many of these TRIMs themselves are inducible by interferon. You know, having like an enhancement loop that would increase the expression of these genes and allow them to have their functions. We then perform a screen on the 70 members of the TRIM family, and we found that a large number of them can have functions, positive functions in regulating signaling pathways. And you can think about this as also having effects indirectly against viruses, because if you are regulating positively the interferon response, an interferon eventually will induce antiviral proteins. So this is something that we have been very interested in looking, you know, how TRIMs regulate interferon and other inflammatory pathways. So this is just an example of how many TRIMs, and there are more to this already, that can have both negative and positive functions in regulation of pattern recognition receptor signaling. So upon any virus or pathogens, many TRIMs can have been shown to have to regulate these pathways, mostly via ubiquitination of signaling factors. One of the proteins that we focused on after our screen was TRIM6, which I'm going to tell you a little bit more about. So for this, again, I want to draw your attention to the kinases TBK1 and IKK epsilon that I mentioned before that are responsible for phosphorylation of IRF3 and IRF7. And for a long time, were known to play redundant roles in phosphorylation of IRF3 and IRF7. So in the absence of IKK epsilon, TBK1 could compensate. But you know, later in 2007, it was discovered that IKK epsilon has a non-redundant role that can phosphorylate one site on STAT1 that helps the formation of this complex, the IHGF3 complex, STAT1, STAT2, IRF9. There are transcription factors. This complex translocates to the nucleus, and it's required for the induction of these interferon-stimulated genes. And SERIN708 helps, phosphorylation of this site helps the formation of a stable complex. But how the IKK epsilon is activated was not known for a long time. And so during these studies, we found that TRIM6 can synthesize these type of polyubiquitin chains, K48 polyubiquitin chains, but these type of polyubiquitin chains in particular are not covalently attached to any protein. So they do not link covalently to IKK epsilon and target it for degradation. What these do actually is to promote protein-protein interactions, so promote oligomerization of IKK epsilon, and this activates IKK epsilon, helps its cross-phosphorylation that leads to its full activation for, so it's able to phosphorylate its substrates like STAT1 or IRF3 and IRF7. So basically, we elucidated a molecular mechanism by which TRIM6 can activate interferon pathways, and we demonstrated that this could be important in controlling, especially influenza virus replication. And I will tell you a little bit more of other viruses that we have been studying. But what is important is that this is an unconventional type of polyubiquitin chain. It's not the regular type of polyubiquitins that are studied. These are difficult to study because they are not covalently attached. So there are a lot of experiments that need to be done to dissect and demonstrate that the chains are not covalently attached, and can lead to different functions. So after this, if we were asking questions like if TRIM6 is indeed a very important host factor in the induction of antiviral responses, then we hypothesized that there might be viruses that would antagonize it. So we started looking for viruses that were already known to antagonize the interferon pathway by targeting specifically IKK epsilon, since this is the substrate that we found for TRIM6. So by asking this question, we could also ask whether the inhibition of IKK epsilon was via blocking the activity of TRIM6. So this part of this project was published in 2016, and this was done by a postdoc then in my lab, Priya Baraj, and it was a huge collaboration with other labs. So the interesting thing is that by that time, our collaborator in Mount Sinai, Ben-Hur Lee, had found that the matrix protein of Nipah virus could inhibit IKK epsilon, and that's how we wanted to narrow down the mechanism. So Nipah virus is a highly pathogenic virus that has had outbreaks in Asia. It has high mortality rates. It has to be. We have to work with it in BSL, in high containment facilities in BSL-4. It can be transmitted. The reservoir is believed to be the bats, and can be transmitted to other animals and to humans. And there have been reports of human-to-human transmission, and it can have respiratory effects and also encephalitis and other problems. But it's a very highly pathogenic virus. So Nipah virus, when it infects cells, like other viruses, releases his genome and allows the genome to replicate. So once the proteins are expressed, this protein that I was telling you about, the matrix protein, it's actually a very interesting protein because it's a structural protein. It was known to be important for budding of the virus. So it's required for the formation of viral particles and release. But something that was not known is that the matrix protein required trafficking. It needed to go to the nucleus and then be transported to the membrane to package the virus. And it was not known what this trafficking meant or why was it needed for the virus. And our collaborators, Ben-Hur Lee, discovered that there was this site K258 that was required for this trafficking. So we use this mutant, you'll see in a minute, because this trafficking is also important for the antiviral, for the antagonism that we discover. So the question that we ask is, since Nipah, the matrix protein, could block IKK epsilon, we wanted to know if it can do it by blocking the binding of unanchored polyubiquitin chains that we found were important for the activation of IKK. So the first thing that we did was to use a luciferase reporter assay. So in this assay, it's a promoter that is linked to a luciferase reporter that can be transfected into cells. And in the presence of IKK epsilon, we can activate this reporter. And in the presence of Nipah, the wild type M, we can see that there is a reduction in the interferon induction. So indeed, it's antagonizing the effects of IKK epsilon. But the use of this mutant, the K258A, that cannot traffic, loses this activity. So actually gives us a good control to test the effects of the matrix protein. So the next assay that we use to test whether this is affecting the interaction of these unanchored K48 ubiquitin chains with IKK epsilon is an immunoprecipitation assay in which we can overexpress IKK epsilon in the presence or absence of the matrix protein and see what interactions occur in the presence of IKK epsilon. So what you can see first is that when you overexpress IKK epsilon, and we can pull it down, you can see in this Western blot that you can detect these non-corporeal ubiquitin chains with IKK epsilon. And IKK epsilon actually, when it's overexpressed, is active. So you can see here that we have high levels of phosphorylated IKK epsilon. And also you can get interaction with IRF3, which is substrate downstream, and the IRF3 that is interacting with IKK epsilon, it's phosphorylated. So now what happens when we overexpress the matrix protein, the wild-type matrix protein? You see that we lose interaction with these non-corporeal ubiquitin chains. This results in reduced phosphorylation of IKK epsilon, reduced phosphorylation of IRF3, but the interaction with IRF3 is not affected. So it's really the activity of IKK epsilon that is affected and by targeting these ubiquitin chains. And we can see that we can recover this activity in the presence of the 258A mutant of the matrix protein, which does not inhibit interferon. So, is it this happening via TRIM6? Is it the matrix protein targeting TRIM6 for this effect? So we went back to this luciferase reporter assay, and what you can see is that if we add in the presence of TRIM6 and interferon, we get an induction. So TRIM6 can enhance the levels of interferon-stimulated genes induced by interferon. And in the presence of the wild-type matrix protein, you can see that this effect is reduced. If we use, again, the mutant matrix protein, we only see minor effects. But the other thing that we realize in these assays is that we lose expression of TRIM6 when we overexpress the matrix protein, suggesting that maybe the matrix protein may target TRIM6 for degradation, makes it or less stable, or something happens to TRIM6 in the presence of the wild-type matrix. So in another way to demonstrate that this happens during viral infection, in collaboration with Alex Freiberg in using high containment facilities at UTMB in BSL-4, generated recombinant viruses that lack the matrix protein. So he called delta M. And as you can see here, so this and this is in a background of virus that expresses GFP. So you can see which viruses are infected because they are fluorescent. So you can see that the cells that are expressing GFP appear to express less amounts of TRIM6 as compared to cells that are surrounding it that are not infected. And you can compare that also with cells that are infected with the delta M, which do not appear to reduce much the levels of TRIM6 expression, again, suggesting that the matrix protein, the wild-type matrix protein, targets TRIM6 for degradation. So for this part, we think that we discover a mechanism by a very highly pathogenic virus in which targets what we describe to be an antiviral protein, TRIM6, via interferon production, and basically targeted for degradation. So the next question was, are there any other viral proteins that are known already to target IKK epsilon that could also have this effect? So we looked at this protein BP35 of Ebola virus, which was described previously as a very well-known interferon antagonist of Ebola virus. And since it had been described before to target RIG-I, TBK, and IKK epsilon, we wanted to know if this could be also an effect via TRIM6. So Ebola virus, I'm not going to talk too much about it, but in terms of, like, you all know, that it's very highly pathogenic. There have been outbreaks in Africa, and it has caused a lot of deaths in Africa. And the latest outbreak happened in 2020, caused a lot of cases and deaths. And again, this virus also has to be worked with in high containment BSL-4 facilities. So BP35 is really studied a lot for his role as an antagonist of the interferon response, but it also plays a role as cofactor of the viral polymerase. So after infection with Ebola and expression of the different proteins of the virus, BP35 can complex with other polymerase proteins to help the virus replicate. So it has different functions, and BP35 could have different functions in terms of interferon and replication. So as I said, probably the best studied role of BP35 is its antagonistic role by blocking both, by sequestering double-stranded RNA from RIG-I and MDA5, and but also targeting IKK and TBK and phosphorylation of IRF3 and IRF7. So we asked this question: can BP35 also target TRIM6? So the first thing we did was to test in immunoprecipitation assay. Again, it's like a binding assay to see if TRIM6 could interact with BP35, and indeed we found interaction of TRIM6 with BP35, but not other proteins like the MP protein of the lymphocytic choronohemolytic virus that also was known to inhibit IKK epsilon. So the next question is, whether TRIM6 has an effect during Ebola viral replication. So we generated TRIM6 knockout cells and tested, the role of TRIM6 first during Ebola virus replication. And what we could see is, as we expected from the role that we described, that TRIM6 knockout cells do not produce much interferon, produce less interferon than wild-type cells during Ebola virus. And this occurs late during infection because, as I said, Ebola virus is very good in inhibiting the response. So interferon is only induced later when there is a lot of virus. But these cells seem to also have some dysregulation because they have the knockout cells express more levels of inflammatory cytokines like IL-6. Cells are dead, they are infected, and they are responding, but it's just they don't induce much interferon. So we expected with this result that we will have, you have less interferon. We were expecting to have more virus, right? Interferon is antiviral. You have less interferon. We expect to have more virus replication in these cells, knockout cells. But actually, it was the opposite. So these TRIM6 knockout cells had less virus replication, suggesting that TRIM6 at least has some role in promoting viral replication, Ebola virus replication. So we decided to test whether this interaction between TRIM6 and BP35 results in ubiquitination of the viral protein, and maybe this is affecting BP35 function. So we use mass spectrometry analysis, and we detected one lysine ubiquitinated in the C-terminal region of BP35, which is known as the antagonist domain of the BP35, with this lysine 309 residue, and we corroborated that this is ubiquitinated. So this is an assay in which we overexpress HI-tag ubiquitin in the presence of BP35 wild type or a mutant, a lysine to alanine mutant that we would not get ubiquitinated on that site. And you can see that these multiple bands represent different molecular weights of BP35 because it's covalently ubiquitinated. So indeed, we can see ubiquitination of BP35, and there is a reduced ubiquitination of BP35 in the mutants. There is still some bands, which means that we probably missed some sites in the mass spectrometry, and there are other sites on BP35 that might be ubiquitinated besides K309. But these mutants may allow us to study what is the role specifically on K309 of ubiquitination. So then we try to confirm that TRIM6 indeed has a role on ubiquitinating BP35, and in this assay, it's an immunoprecipitation again, in which we pull down BP35, but in the presence or absence of TRIM6. And you can see in the presence of TRIM6, we can see enhanced ubiquitination of BP35. If we use the knockout cells and we overexpress BP35 and pull it down, we can see that in TRIM6 knockout cells, there is reduced ubiquitination of BP35. So indeed, TRIM6 plays a role in ubiquitination. So then we set up an assay. This is without virus. This is a reporter assay, luciferase reporter assay, mini genome, using only the polymerase components of the virus. As I said, BP35 has a function there. So by using BP35, we can test whether the wild type and the mutant in the presence of TRIM6 would affect the polymerase activity. And you can see that when we add TRIM6 to this assay, we have an enhancement of the polymerase function of the virus. And when we add a catalytically inactive form of TRIM6, we do not see this enhancement. And if we use this mutant of BP35, we also do not see the enhancement. So really, this suggests that TRIM6 ubiquitinates K309 on BP35, and this is important for polymerase function. We also generated TRIM6 knockout mice and we are doing experiments with these mice now. But the first thing that you can see is that indeed, as we see in cells, this TRIM6 knockout mice is a little bit more resistant to Ebola virus infection. And this is probably due because indeed there is less virus replication in some tissues in the TRIM6 knockout mice. I have shown you two very highly pathogenic viruses that can target TRIM6. So now I want to talk about this. We are still investigating the mechanisms of, at the molecular level, how BP35 ubiquitination affects replication, and this is work of a graduate student in the lab, Sarah, and we hope to have a manuscript soon for this. So now I want to shift to another virus, completely different virus. This virus, Zika virus, you probably heard about it in the past years. Now with COVID, this basically there is no attention on this anymore, but it doesn't mean that it's not circulating around. The interesting thing, or the problem that we had with Zika, was that it's transmitted by mosquitoes, but it was found in pregnant women to affect pregnancy, and it was associated with microcephaly in newborns. So the big question is why the virus likes to replicate so well in reproductive tissues, and why is it causing these effects? So we turn to see if anything related with the ubiquitin system would be involved in this. So the way we did this was similar to what I talked about for Ebola, which is that we use infected cells, and we perform mass spectrometry analysis from cells infected with different flaviviruses for comparison, and Zika virus here, Zika B, and we found different viral proteins that were ubiquitinated. Here I'm showing you the envelope protein of Zika virus, which was of real interest for us. And we found two different sites that were ubiquitinated on Zika virus. One that is conserved in other flaviviruses, which this is lysine 38. And the other one is non-conserved in other flaviviruses, this lysine K281. So we wanted to know what the effects of this ubiquitination, and whether they have any relationship with tropism or with the effects that are seen specifically in Zika. We focus on envelope really because it's a really important protein. It's the main component of the virion, and it covers the envelope, and it's important because it's recognized by receptors, and it's important in entry. This was work that we published recently, Maria Giraldo in the lab, in collaboration. This was a huge collaboration with people inside UTMB and outside, especially Peiyong Shi at UTMB. And what we did is first is generate recombinant mutant viruses that have an arginine instead of a lysine in these two sites, that would are the closest possible amino acids to lysine, but they do not get ubiquitinated. So you conserve as much as possible the structure of the envelope with this change, but the arginine cannot be ubiquitinated. And as you can see here, again, with overexpression of HA ubiquitin and infected cells, you can see that the wild type envelope from wild type virus, we can see a smear that corresponds to ubiquitinated envelope, but the other two viral mutants lose ubiquitination. We tested this in cells, like how does this virus replicate in different cell types, and we can see that placenta-derived cell line, where the virus replicates very well, these two viruses are attenuated. With the K38 was the highest effect, but this is a cell type specific effect. So for example, in testes, the K281 didn't have much effect, but the K38 are still was very attenuated. And when we look at mosquito cells, the virus also replicates very well in mosquito cells, of course, because it's transmitted by mosquitoes, but we didn't see any difference there in these mutant viruses, suggesting that this site and potentially ubiquitination in this site is not important in mosquito cells, but it is important in certain cell types in humans. So then we tested the effects in vivo. We infected mice with these viruses, and what you can see there in red, the red line, is the K38 mutant virus, that it's attenuated. It basically causes less disease in these mice. So wild type virus can makes the mice lose more weight, and the K21 didn't have much effect. So these effects translate in survival. So the wild type, we get some mice that die, but the mutant viruses survive. So we looked at replication in different tissues, because this is in the end the question that we had, is if this affects tropism. And it was interesting to see that some tissues like the brain and testes, where the virus replicates very well, the K38 was reduced in virus replication, while the K21 was not. So we looked at more tissues, focusing only on the K38. And interestingly, we saw only differences in tissues where the virus replicates very, very well, like brain and reproductive tissues, but not in other tissues. So really, this suggests that the virus, the ubiquitination on K38, it's important for tropism. And we did experiments using live mosquitoes after blood meal, and we didn't see any difference in mosquitoes. So again, this site does not appear to be important in virus replication in the mosquito. So how is this happening? We obviously had to go back and look at them at which one is the E3 ligase that is doing this. So to cut a long story short, we found, after looking at multiple knockdown screens and looking at different potential E3 ligases involved, we found this one, TRIM6, that it seems to be highly expressed in placenta and reproductive tissues and could be one of these targets. So we made TRIM6 knockout cells, and we can see that there is a reduction of virus replication in TRIM6 knockout cells. And when we use this K38R virus mutant, it really shows a similar effect to the TRIM6 knockout cells. So this really suggests that TRIM6 it's important for efficient virus replication in these placental-derived cells, and that you need an intact lysine K38 residue for this replication. We tested interaction with TRIM6 and in infected cells, and as we can see here in this immunoprecipitation assay, TRIM6 can interact with envelope. We then wanted to confirm that TRIM6 ubiquitinates directly envelope, so we established an in vitro ubiquitination assay. In this assay, what we do is that we used all the components of the ubiquitination system. So we use an E1 activating enzyme. We used this E2 ligase UBCH5A, which was previously already described to act in conjunction with TRIM6. So we didn't discover this one, it was already reported. And we purified TRIM6 to add it to this assay. And we use the wild type and the mutant envelope proteins to demonstrate this in vitro. So in this assay, in the presence, so what you can see here is that when you add TRIM6 to this assay, we can get this shift in the molecular weight of envelope, which corresponds to bands that appear for ubiquitin when we're using an ubiquitin antibody. So basically, again, when you get ubiquitination of envelope, this is covalent ubiquitination. So then you have a shift in the bands upwards, and you see the more TRIM6 is added, the more you get increase in these molecular weight bands that represent ubiquitinated envelope. So what is, as a control, we can see that if we use this catalytically inactive TRIM6, so it's a deletion of TRIM6 that lacks the ring domain, we do not get any increase in ubiquitination. We also need ATP. So in the absence of ATP, this reaction does not happen. We need energy source as an energy source ATP. And what we can see is that in the presence of the mutant K38R envelope, we have a reduction of ubiquitination, as we would expect, but it's not completely gone. This is probably due because of the presence of the other side, the K281 side, that is still there, probably still gets ubiquitinated, and we can still see some envelope ubiquitination, but the reduction is it's clear. And we can see that in the presence of the K281 mutant almost completely loses ubiquitination. So this really, what indicates is that indeed, in vitro, TRIM6 can directly ubiquitinate envelope, and in these two sites, in K38 and K281, and that they both probably play a role and are required for full ubiquitination of envelope. So then we wanted to test if this has an effect in vivo. So we generated TRIM7 knockout mice to test whether TRIM7 has a role in virus replication and whether it will have a role in tissue tropism. And so what you can see here really nicely is that in TRIM7 knockout mice have reduced virus replication, again, in the tissues where the virus likes to replicate, like brain, testis, and uterus. Interestingly, here we also see a defect in the eye, maybe because it also has a connection with the brain, and maybe that's one of the reasons why we also see a difference here. But we do not see difference in any other tissue. So really, again, indicating that TRIM7 and this ubiquitination may occur in a tissue-specific manner, and it suggests really that TRIM7 could be a determinant of tropism for Zika virus. So this is all very nice, but we want to know a little bit more about the mechanism and how is this occurring. And so ubiquitination happens in the cell. All the machinery is obviously inside the cell. I'm not showing you here, but in some experiments, we could see that during infection, we could find some relocalization of TRIM7 to some compartments, some vesicles that seem to be associated with the Golgi, and suggesting maybe ubiquitination may happen during viral replication at later time points when the virus is maybe being formed and it's being transported to outside the cell. But most of the ubiquitination has been studied inside the cell. Like, there's really not much evidence that viruses come out of the cell with ubiquitin and that ubiquitin plays a role. There are maybe only examples with influenza of this happen, but we wanted to know if this could be something that plays a role. So we hypothesized that maybe the reason why we're seeing TRIM7 in the Golgi and that the virus gets ubiquitinated in the envelope, and the envelope has a very important role in entry, we wanted to know if the virus that is released contains any ubiquitin. So for this, what we did was to collect supernatants from infected cells with Zika, and we clear them, we concentrate them, and clear them as much as possible from contaminants. And what we did was to use a specific anti-K63 ubiquitin antibody for an immunoprecipitation of virions. If these virions contain some ubiquitin, we would be able to isolate these virions that contain ubiquitin. As a control, we also use an antibody against the envelope protein to see how much envelope we can pull down. So what you can see here in the left is that when we do this immunoprecipitation, either with the envelope antibody and we probe with an antibody against envelope, we can see the band that corresponds to non-modified envelope, which is approximately 48 kilodaltons, something like that, right below 50. But then we see this smear of bands that go up, all the way up, that correspond to possibly multiple chains of ubiquitin. And if we compare these with the immunoprecipitation with the anti-K63 antibody, we can see that the high molecular weight bands really correlate with ubiquitin. And if we then reprobe this blood with anti-K63 ubiquitin, we can see that these bands really correlate with the high molecular weight bands of envelope, really suggesting that the virion itself contains some amount of envelope that is ubiquitinated. This doesn't seem to be complete. It's just a proportion of envelope that seems to contain ubiquitin. We did something similar, like collecting supernatants from cells from wild-type and TRIM7 knockout cells to see if TRIM7 indeed is responsible for this. And we could see that there was a loss of ubiquitin in the virions that were collected from TRIM7 knockout cells. So how else can we do this so we can really show it? How can we more visually show it? So we use this anti-K63 ubiquitin antibody, and then we use a secondary antibody that is labeled with gold particles, and we perform cryo-EM to try to see the actual viral particles, to see if we could recognize, ubiquitin around the virion. So what you can see here is an example of one of the particles that we could see. So the little very dark dots that you see around the virion are gold particles that are recognizing the anti-K63 ubiquitin antibody. So what is interesting about this is that the first thing is that not all the virion is covered with these particles, which suggests that not all the 180 units of an envelope that are contained in the virion are ubiquitinated. And the second is that we found like a small proportion of the viral particles that contain these gold particles. So maybe about 15% from the preparation, we could see that had viral these gold particles. So I think this also demonstrates in a more visual way that the virus can contain some ubiquitin, and we can use this K63 antibody to recognize it. So if the antibody works well, we also wonder if this antibody would be neutralizing. This anti-K63 ubiquitin antibody, if we mix it with virus, would it be neutralizing? Would it block virus replication? And indeed, what we see is that if we add increasing amounts of this antibody, we can reduce the infectivity of the wild-type virus and the K281 mutant virus, but not the K38 virus that is in red. So this, again, strongly suggests that indeed the antibody can recognize K63 ubiquitin chains and reduce infectivity of the virus, but only, you need that K38 site intact. We use as a control an anti-ubiquitin antibody as K48, and we saw very little effect. We also wanted to know if this happens in vivo. So we administer mice with this anti-K63 ubiquitin antibody and infected mice. And what we could see is, again, that this antibody could reduce the levels of virus replication in at least in the serum when we looked at virus replication in the presence of this antibody. How is this occurring? So the virus contains ubiquitin. So is it that why does it need ubiquitin, the virus? Why the virions would contain ubiquitin? So the next obvious question would be if this has any relation to attachment. So we perform an attachment experiment in which we incubate cells at 4 degrees with wild-type or mutant viruses. We let them attach for 30 minutes at 4 degrees, and then we collect RNA to measure virus attachment. And what we can see is that indeed wild-type virus can be attached to the cells, and the K38 mutant virus is highly reduced in the ability to attach to the cells, with just not very significant reduction in the K281. And if we shift to 37 degrees, we allow the virus to go inside the cell, and we can see that indeed, because there is less attachment to the cell with the K38 mutant, there is less entry. So you can detect less virus entering the cell with these mutant viruses. Finally, we wanted to know if this is related to binding to a receptor. There are a few different receptors that have been described for Zika, but they are not. It's not clear which one is the most important. There is still a lot of controversy, but we use TIM1 to test whether this would have an effect in attachment attachment to the receptor. So in this assay, we bind TIM1 to the beads, like if it was an immunoprecipitation assay, and we mix it with virus, wild-type or mutant virus, and what we can see is that the wild-type virus can interact very well with TIM1, but the K38R loses this interaction with TIM1, suggesting that at least TIM1 has some affinity for virions that contain ubiquitin. So to summarize this part, so what we think that is happening is that, the mosquito transmits Zika to humans. Mosquito does have an ubiquitin system, but it doesn't contain the same TRIM7 that the humans have. So it's possible that ubiquitination doesn't happen in the right place or in the right chains. and in mosquitoes it's not important. But once the mosquito bites and the virus infects humans, the virus is disseminated, it goes to different tissues, and when it finds the right tissue where TRIM6 is expressing, then the virus starts replicating better. And this is because there is a stronger interaction of viruses that come out from tissues that contain TRIM6, because it contains ubiquitin. So this gives an advantage to the virus to attach better to the receptors and replicate better in these tissues. Now, it's not like the absence of ubiquitin renders completely inactive the virus. So the virus can still replicate, but it's just has weaker interactions with the receptors. The interesting thing is that TRIM6 has been reported to have other functions like antiviral functions. So it's interesting that TRIM6 is having different functions during replication. Different viruses, it can block norovirus replication, but for Zika at least, we showed that it's important in ubiquitination of the envelope and allowing some tropism to occur. So in the last two slides, I cannot ignore COVID. Obviously, this has been a huge problem. We all know about it. What it has been really, we have learned a lot in the last year and a half on how inflammation causes disease in patients, and how so the virus infects epithelial cells, but there is recruitment of immune cells that cause damage to the tissue, and this is due to really dysregulation of the immune response and expression of cytokines. Especially type 1 interference should be beneficial, right? They should be blocking virus replication, but they also have an inflammatory role. So there has been a lot of research trying to figure out also antagonists of the interferon response. So in collaboration with Pei and Shi, we did a screen, and we found multiple viral proteins of SARS-CoV-2 that could block at very different and multiple levels of these signaling pathways to block the antiviral response. We interestingly found and focused on one of these proteins, the ORF7A, which is an accessory protein that is not very well known what it does during viral replication, but it's known to be a membrane, possibly a membrane-associated protein that it may be involved in immune dysregulation. And what we found is that this ORF7A, it's ubiquitinated. In the panel A here, you can see that it's highly ubiquitinated. And making mutants, we found this lysine 119 that has reduced ubiquitination. And when we use this in the type 1, in the reporter assays that I mentioned before, we can see that while the wild-type ORF7A can block interferon signaling, there is the mutant has less ability of block this interferon response, suggesting again that ubiquitination of this ORF7 can also affect how ORF7 antagonizes the response. So with this, I think I have shown you, there are multiple viruses that are affecting, are interacting with the host response. So the ubiquitin system has a huge role in maintaining homeostasis and regular functions of the cell, disposal of unwanted proteins in the cell normally. Like you can see on the right that you can think this as TRIM6 and TRIM7 having normal antiviral roles, having effects on antiviral in immune signaling and other types of signaling that maintain the host healthy. But then you have all of these other effects on the left side that the viruses are interacting with these factors that are normally antiviral, and either destroying them, like the example of M and TRIM6, or taking advantage of them, hijacking them to allow ubiquitination to help them to replicate. So with this, I'll finish, and I'll thank multiple people that have been involved during the years in all of these projects, the people in my lab that I mentioned during the talk, collaborations. This work would not have been able to be done without the collaborations of all the people in BSL-4, Alexander Freiberg, Alexander Bukayev, Pei-Yong Shi for all the Zika and CoV-2 experiments. And there were a lot of collaborators that helped making the TRIM7 knockout mice, Sonja Best, Wendy Murray provided mice, team knockout mice, Ben Hurley for the Nipah virus, and I'll finish and take questions if there are.
Thank you so much, Dr. Rajsbaum, for that informative presentation. And we will now begin the live Q&A portion of this webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So it looks like we already have some great questions coming in. Our first question, what determines the type of linkage of the polyubiquitin chains, and what makes K48 or K63 polyubiquitin chains?
Great, thanks for that question. So this is actually an interesting point. As I mentioned, there are about 40 E2 conjugating enzymes encoded by the human genome, and in general is the E2 conjugating enzyme, the one that is responsible for making the type of polyubiquitin chain. So you need the association or the interaction of the E2 enzyme with the E3 ligase, and the E2 is the one that it decides where to put that ubiquitin on the other ubiquitin to make the polyubiquitin chains. Some of them are promiscuous and can make different types of polyubiquitin chains, but there are some of them are more specific and can make only one type of polyubiquitin chain. So it's usually the E2 conjugating enzyme, the one that decides what type of polyubiquitin chain is made.
Thank you so much. And we have another question coming in. Why does the virus require ubiquitin to enter the cell in the case of Zika virus? And do other viruses also contain ubiquitin that helps them enter those cells?
Yeah, so this is also an excellent question, and this is something that we have been studying just lately, so it's not really much known about the mechanisms or how or why the virus would want to have ubiquitin on the virion. So as I said, for Zika virus, the ubiquitination of the envelope protein in the virus is not really required for entry, but it helps the virus to enter the cell. So what we think is that having the presence of some ubiquitin chains in the virion on the envelope protein, this helps enhance the affinity of the envelope protein to interact with the receptors. In our case, we tested TRIM1, so somehow we don't know exactly at the molecular level, but the interaction of ubiquitin with the receptor helps the envelope binds better. And we do know so far at least one other virus that can contain ubiquitin in the virion. This is influenza that has been reported that can contain inside the virion non-covalently attached ubiquitin, but this apparently helps the virus in a completely different way to Zika. Apparently, when influenza virus enters the cell, this ubiquitin helps the uncoating of the virus to release the genome in the cytoplasm. And so far, there are no other viruses that we know of that contain ubiquitin, but we do think that other viruses might contain ubiquitin that may help them enter the cell, and it's difficult to study, so that's probably why people have not found them.
Dr. Rajsbaum, this has been a great presentation. Thank you so much for your time and for your important research. Would you like to provide any closing remarks for our audience before we go?
Yeah, so I mean, thank you all for attending this session. I think one of the points I want to make is that, yes, we are in COVID time and we are all really paying a lot of attention and more attention on viruses, but there are other viruses out there that we are not paying attention right now. And I don't know if anybody has heard, but there is an outbreak right now in Asia of Nipah virus. A lot of people think that this could be potentially a future pandemic, even though this virus is really highly pathogenic and kills people. So maybe makes it more difficult to transmit. But we should continue paying attention to other viruses that are out there. I think something came in.
Sure, we can throw in one more question. This question says, I'm not sure if you're aware, but the Zika virus protein are made as a polypeptide translated into the ER. Is it possible that what you are observing with loss of ubiquitination of the glycoprotein is a change in the trafficking of the immature GP and release of aberrantly glycosylated GP, which alters the interactions with leptin binding proteins, such as what you've shown?
Yeah, I mean, thanks for this question. This is a great question. And obviously, when we were doing these experiments, we were concerned about this, that ubiquitination does occur inside the cell. And we do think that actually where the region in the cell where this is occurring is somewhere between the ER and the Golgi. So yes, there could be a possibility that the ubiquitination would be altering trafficking, processing, or release of the virus. But actually, we tested this during our experiments, and we did not see in the mutant viruses any difference in the release of the virion compared to wild-type viruses. So we do not think that the ubiquitination on the envelope is affecting, at least in major, doesn't have major effects in the release. Ubiquitination of the envelope could have other effects during the replication of the virus, also depending on which of the two sites are ubiquitinated, if the K38 or the K281. So there is still a possibility that there are some effects in trafficking and in release, but we do not think that the mutant virus, the K38R mutant virus, has a defect in trafficking or how the virions become mature and are released. I hope that answered that question.
Dr. Rajsbaum, thank you so much for your time again today and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions and questions we didn't have time for today. And those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast will be viewed on demand for 2 years until September 9th, 2023. Laboratories will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and stay healthy. Bye-bye.